My presentation concerns the potential effects of phytocomplexes studied in the field of virology and of immunology towards this pandemic and for their potential application basing on results that were obtained. Until 2019, 6 coronavirus capable of infecting human were known. Two of these, both from the beta coronavirus group, caused major epidemics characterized by fatal respiratory disease. SARS-CoV, responsible for the severe acute respiratory syndrome in 2002 in the province of Guangdong in China, and MERS-CoV, responsible for the Middle East respiratory syndrome in 2012 in the Kingdom of Saudi Arabia. Recently, a new CoV called severe acute respiratory syndrome coronavirus 2 was responsible for the outbreak of COVID-19 pandemic on the 31st of December 2019 in China. The previous SARS and MERS outbreaks involved 29 and 27 countries respectively. And given that over 170 countries have been affected by COVID-19 pandemic after only 3 months, the severity of the current infection is evident. The most common symptoms and signs of SARS-CoV-2 infection are respiratory symptoms, fever, dry cough, fatigue, dyspnea, where the clinical manifestations of infection range from asymptomatic to severe pneumonia. It is noteworthy that pneumonia, a lower respiratory tract infection, has occupied the first position in the communicable disease rank and the fourth position in the top 10 causes of death globally. So that's a very important point. Other mild symptoms are characterized by gastrointestinal ones, such as diarrhea. And, there is also a very important part of symptoms involving cardiovascular system. Well, the actual therapies that were tried for coronavirus treatment include the nucleoside analogs, for example, remdesivir, favipiravir, and all the ones that you can read on the slide. The protease inhibitors, such as lopinavir, danoprevir, rabidol, and other drugs were tested, for example, chloroquine, hydroxychloroquine, and others focusing on immune system. I think that the most used one is dexamethasone, that is a corticosteroid, and also monoclonal antibodies such as tocilizumab and others. Also, the plasma therapy has been tested and it seems to give encouraging results. And I think it's a good point. Well, in the present conference concerning my presentation, I will talk about food components, phytocomplexes that may be helpful in managing coronavirus disease 2019. There is an untargeted approach that is based on immune system enhancement through food components, phytocomplexes and isolated compounds, and this is being investigated in several trials, and a targeted approach that is focused on the interaction between specific compounds and the host or various proteins, and is being investigated by in silico studies and also in vitro studies. Infection by SARS-CoV-2, the virus determining COVID-19, is characterized by the binding between viral spike protein and angiotensin converting enzyme 2, namely AC2. The activation of the spike protein is mediated through proteases such as TMPRSS2, which play important roles in the viral infection. Infected transmembrane serine protease 2 cleaves SARS-CoV-2 as protein and mediates its entry into the cells. After entry, followed by endocytosis, coronavirus infection causes PAK1 upregulation, a kinase that mediates lung inflammation, lung fibrosis, and other critical mortality factors. SARS-CoV-2 infection is associated with increased levels of chemokines and activated pro-inflammatory cytokines that lead to development of atypical pneumonia with rapid respiratory impairment and pulmonary failure. A retrospective observational study found higher serum levels of pro-inflammatory cytokines such as interleukin-6, interleukin-1, TNF-alpha, and others in patients with COVID-19 compared to individuals with mild disease. Type 1 interferon production is impaired and severe cases leads to ARDS and widespread coagulopathy. COVID-19 pathophysiology is initiated by SARS-CoV-2 gene products, among others, NSP1 and ORF6 proteins, leading to a catastrophic cascade of failures. These viral components induce signal transducer and activator of transcription 1 dysfunction and compensatory hyperactivation of state 3. In SARS-CoV-2 infected cells, a positive feedback loop established between state 3 and plasminogen activator inhibitor 1 may lead to an escalated cycle of activation in common with interdependent signaling networks affecting COVID-19. Specifically, PAA1 upregulation leads to coagulopathy characterized by intravascular thrombi. Furthermore, a reproduction of PAI1 leads to its binding to TLR4 and macrophages, inducing the secretion of pro-inflammatory cytokines and chemokines. So, as you can see, there are many molecular networks that are involved in the inflammation induced by this virus. Traditional herbs from different geographical areas are considered potential sources of new molecules for the treatment of viral infections. They could also be the basis for identifying nutraceuticals useful as adjuvants in the treatment of COVID-19 infection. Over 200 of phytocomplexes have been tested against SARS-CoV, and those obtained from, example, Licorice radiata, Artemisia annua, Pyrosa, Lindera aggregata have shown a significant antiviral action with effective concentration in the range between 2.4 and 88.2 microgram per milliliter. You know, there are some cases where the phytocomplexes seem to be in vitro really active. Propolis has been tested against various viral disease organisms. Initial success have prompted research to determine the most useful components which may be modified to produce more active and specific pharmaceuticals. For example, viruses that were controlled by propolis in preclinical models include influenza, herpes simplex virus type 2, and HIV in vitro, in preclinical model, of course. Well, chemical analysis revealed the presence in several exons from propolis of a wide range of organic molecules that are very different. These include flavonoids like, for example, aspartame, myriacetine, galangine, pinosandrine, quercetin, camphor, and also other classes of phytochemicals such as limonene, caffeic acid, and its phenyl ester. Also, limonene, for example, has been observed to be active against some viruses. Well, in silico studies outlined the potential of some phytochemicals found in propolis to inhibit ACE2. This is the case of hydroxycinnamic acids, including caffeic acid, coumaric acid, cinnamic acid, and some flavonoids like, for example, rutin and myriacetine. You know, limonene, quercetin, camphor show a potential to inhibit, for example, a polymerase that is involved in viral replication. So these isolated compounds may be further investigated. The main protease of coronavirus SARS-CoV-2, named MPRO, is essential for coronavirus processing of polyproteins and for its life cycle. And a fair inhibition of the active site of this enzyme is relevant target for drug discovery. Caffeic acid, phenyl ester, galangine, trazine, and caffeic acid itself that are found in propolis seem to be able to have the potential to inhibit this enzyme. So they may be further investigated. Further mechanism of camphor consists of inhibition of the 3A channel of SARS-CoV encoded by ERF3A of SARS-CoV, and to determine downregulation of transmembrane proteases serine-2, that is a protease that is important for cleaving the protein named spike protein of the virus. In several experimental models, the ability of caffeic acid and its phenylester to downregulate PAK1 induced by SARS-CoV-2 infection may be relevant. As PAK1 activation include lung fibrosis, which is an aggravating factor in COVID-19, and PAK1 inhibitors may help both combat the virus and restore normal immune response, these propolis compounds may be considered for further studies to test them as coadjuvant in the treatment of this pathology. The same active principles are able to hit target of other molecular networks, producing actions of inhibition towards inflammation and towards oxidative stress, determining an increase of humoral and cellular immunity. You know, these actions are the result of a modulation with different active principle on different molecular targets. So, you know, if we look at their potency towards single targets, it is generally lower than that of pharmaceutical tools used as a comparison. But what we can find with this kind of phytochemicals and phytocomplexes is the wide range of activities. So that is the very interesting point. In preclinical studies, propolis promoted immunoregulation of pro-inflammatory cytokines, including reduction of interleukin-6, interleukin-1, and TNF-alpha. This immunoregulation involves monocytes and macrophages, as well as JAK2, STAT3, and FKB and inflammasome pathways, reducing the risk of cytokine storm syndrome, that is a major mortality factor in advanced COVID-19 disease. Propolis has also shown promising as needed in the treatment of virus and the comorbidities that are particularly dangerous in COVID-19, for example, those concerning vascular trouble, because the molecular mechanisms that were investigated suggest its usefulness also in this kind of pathologies. Well, we said that SARS-CoV-2 entry into target cells requires spike protein binding to ACE2 activation by TMPRSS2. After binding, several signals are triggered, allowing viral endocytosis and PAK1 activation, which reduces the adaptive immune response and antibody production against virus. PAK1 also stimulates CCL2 production, which generates a fibrotic response. Viral infection induces nucleotransition factor and FKB activation, generating local pro-inflammatory cytokine production. Propolis-derived compounds then regulate the expression of TMPRSS2 and the anchoring ACE2, which limits the end of the virus. Furthermore, they promote NFKB and one-side macrophage immunomodulation, and there are also other effects that may help fighting SARS-CoV-2 infection. Well, in this context, we will focus on phytocomplexes and phytochemicals that were tested for their effects, at least in vitro experimental models, and that may be useful for further investigation for helping treating COVID-19 affected patients. Well, we talk about a virus targeting approach and a host targeting approach, because often these phytocomplexes are able to determine effects toward the host, because, for example, they can modify immune system, but also they have some components that are able to eat up viral proteins, so they can do both. In silico studies suggest the ability of the reported molecules to affect the SARS-CoV-2 protease. This approach brings together in silico analysis and chemical characterization analysis of phytocomplexes, and it may provide novel compounds able to act directly on viral enzymes and proteins. These colleagues focused on African plants, for example, And they applied this in silico approach in order to screen many alkaloid and terpenoids. So according to the experimental model, these two molecules that you can see on the slide seem to be the best candidate as potential inhibitors of this coronavirus chymotrypsin-like protease. Okay, let's focus on another culture, medical culture. So let's focus on traditional Chinese medicine. Shuangguang preparation, a Chinese traditional patent medicine, also called proprietary Chinese medicine, used for the treatment of acute respiratory tract since 1973, is a classical purified herbal preparation extracted from three Chinese herbal medicines, namely Lonicera japonica, Scutellaria bicalensis, and Forsythia suspensa. In particular, Shuangguang formulation, its three source herbs and several compounds isolated from these herbs in vitro showed antiviral activities against HIV, adenovirus, influenza virus, EV71, but the mechanisms of action of this substance remain large unknown. But in vitro results are very encouraging. The oral liquid showed concentration-dependent inhibition of this SARS-CoV-2 protease, and the resulting SC50 values were very interesting for a preparation, and it changes depending on the pharmaceutical company producing it. But this data show that in vitro, Schwann ganglion preparation have an inhibitory effect towards this protease that is very important for viral life cycle. You know, there was also an investigation about one of the main compound, and in particular, authors focused on bicaline. And this compound was shown to strongly inhibit SARS-CoV-2 protease, and it is also very interesting because it is very strong in this kind of effect. Well, another kind of experiments were exerted in order to understand, once they could understand that it works against the protease, if it's able to reduce viral titers. Well, lab preparation and compounds were tested against a clinical isolate of SARS-CoV-2 in various cells. And the three oral liquids all showed concentration-dependent inhibition of SARS-CoV-2 replication. And the resulting of massive effective concentration were very interesting. We are in the range of 1.5 microliter per milliliter respectively. And it's very interesting. Okay, another case, oleander, is a tree originating from Asia and has been the subject of several studies and experiments in various fields of medicinal chemistry and medicinal science. You know, some colleagues chemically characterized an extract of its leaves and isolated oleandrin, a cardiotonic glycoside responsible for the poisonous of the sap of the plant. The authors test the antiviral activity of this phytochemical. In vitro, on various CCL81 cells, this glycoside inhibited viral replication of SARS-CoV-2 at a concentration of 0.05 microgram per milliliter. The data presented here demonstrate the ability of oleandrin to inhibit viral replication when inoculated before infected cells, and that means a potential preventive effect. But it's not the only thing that we're going to see for this isolated compound. In fact, in the same work, the effect of 22 CL cells with oleandrin was evaluated 24 and 48 hours after infecting them. The results showed that oleandrin reduces viral titer in this condition, outlining a potential application when the infection is already present. Well, we have some data for timing. I can't show you all phytocomplex tests in this context, but I want to show you this preparation based on echinacea and published it recently. There is an in vitro study showing the ability of an echinacea-based preparation to inhibit SARS-CoV-2 in addition to other viruses studied in the same work. Once again, this is a virus targeting setting. With these phytocomplexes, is it possible to hypothesize host targeting as well? Okay, let's have a look to this potentiality. Well, several extracts from this plant were shown to affect a plethora of molecular networks, resulting in a potential reduction of inflammation. So we have, we can say that there are two effects. One is virus targeting. The other one is host targeting, and this is very important. So this is what we can see in this case. Well, in Italy, several people talked about naringenin. Well, there was a big noise around this molecule. You know, it's a flavonoid, for example, found in orange, for example, in orange juice, but also in orange extracts, in orange peels extract, we have a lot of this compound. Well, naringenin showed a strong antiviral activity in vitro against SARS-CoV-2. Interestingly, besides its antiviral power, naringenin has anti-inflammatory activity by inhibiting TNF-alpha and interleukin-6 secretion, which may synergistically enhance its antiviral effect in vivo. I mean, the inflammatory effects of the virus in vivo. The potential possible naringenin mechanismatic treatment is that it could tackle both viral infection and cytokine release, cytokine storm syndrome in COVID-19. So what I wonder is why, as a scientist, don't focus on these compounds that are very promising for their effects, and generally they have a very low toxicity. It would be very interesting to test their effects from a clinical point of view in order to understand if they can help treating infection or not. What I say is that for most of these compounds is to find a way to get them more bioavailable and to find a way to find a way they can rest more time in the body when they're ingested. So that's the point. But what I could observe is that with all the compounds that I showed you and the phytocomplexes that I showed you, there is a paradigm shift from the one target, one drug to network target multi-components. So it's an evolution of great importance to which we witnessed in recent years. For example, we could observe that some compounds some compounds from vegetal matrices and also some phytocomplexes are able to also modulate the contractility of the smooth muscle of respiratory tract. And also this is very important. And that's what we do in our laboratory research. So I want to invite all of you to think about all these things and also to collaborate with us, because what we do is testing phytocomplexes against the respiratory tract smooth muscle to we can understand which molecular mechanisms are involved in this modulation, and we can also say that while phytocomplexes affect several parameters concerning inflammation, and while they do also antiviral effect, they can also modulate their respiratory tract. So I want to thank you for kind attention, and I'm here for questions. Thank you for your kind attention.
Audible? Can you hear me?
Yeah, can you see the question, of course?
Yeah, just that was a very nice, interesting presentation, sir.
Thank you.
I would like to know, like, based on your experience, or did you come across any study relating to predictors of interleukin-6? Like, can we predict patients early that they would go in for a high levels of interleukin-6? Any kind of studies are being done, or did you find any predictors or predictable markers for rise in IL-6 and other LDH or serum ferritin levels? Are, because those kind of markers are very important for the patients to develop cytokine storm. So any kind of predictor studies are going on, or do you know any kind of predictors that we can use in our patients to find that they might land in respiratory complications a little bit earlier? What's your comment on it, sir?
Okay, thank you for your question. You know, the predictors that are often used are the cytokines that lead to inflammation. For example, interleukin 6, interleukin 1, TNF alpha. And as you know, normally they are not revealed during normal exams of blood. However, we can, during research, try to see what happens with our substances in vitro models. Of course, I don't know if, but we may also do ex vivo experiments, for example, taking blood from patients and see how they react to spike protein, because there are some treatment that may lead to a reduction of the production of pro-inflammatory cytokines when we took the inflammatory cells from the blood of the patients. And if this patient have cells that produce low amounts of interleukin 6, for example, I can imagine that they will not undergo severe conditions. But you know, I have to try that, and I haven't done it that. So, but it's an idea that we can work on.
Thank you so much, sir, for your answers. Thank you.
Okay, you're very welcome.